It's my pleasure to bring Rebekah Rogers here to give a seminar in our department, the first of the series for fall. I have known Rebekah since she was an undergraduate at the University of Georgia. She was a foundation fellow in the Department of Genetics and got her PhD in genetics. She did some work in the lab where I did my postdoc, Jeff Benetson's lab. She was working on, I think it was strawberry? Yes. Fragaria? Okay, so she did some work on genome dynamics of strawberry. And then from there went to grad school at Harvard with Dan Hartle, where she worked on chimeric and duplicated genes and how that might lead to rapid adaptation in Drosophila. She did a postdoc with Kevin Thornton as well as Monty Slatkin, and then joined UNC Charlotte in 2017 as an assistant professor, and then just recently in 2022 went through tenure. So with that, I'm just going to pass it over to Rebekah, and let's thank her for coming.
Great. So I'm Rebekah Rogers, and like you said, I'm on. Green. Does that work? Cool. Okay, I thought we were ready to go. All right. So if this talk runs a little bit over, someone can give a yell and let me know to skip a few slides. And if it runs under, I'll tell you about all of our trials and tribulations trying to get long reads for these freshwater mussels. All right. But we're using genome sequencing to study evolutionary genomics. All right, so I'm Rebekah Rogers. I work in the field of genetic novelty, and we study how you get brand new gene sequences that help animals adapt to new environments. And we spend a lot of our time studying mutations like these that copy and shuffle pieces of DNA around the genome, and they change large chunks of DNA at once. And they're really good at creating brand new gene sequences where there weren't any genes before. So these mutations can pick up a piece of DNA and move it to a new location, which oftentimes is associated with a change in gene expression for that gene. They can copy a gene, they can stick it in the genome a second time, which gives you spare parts that can be tweaked to generate new functions. You can mix and match pieces of genes together to create regulatory changes and new gene formation. And you can also create something similar with de novo genes, where the 5' end of a gene lands next to an untranscribed sequence and creates a new gene like nothing that's ever existed before. So we know that these mutations can create changes in gene expression, changes in gene content, and changes in gene organization. And so we're studying how they play a role in adaptation and generate novelty. So this is the substrate of variation that's present that then natural selection can act upon. A lot of these mutations do things that are absolutely useless. Some of them may be detrimental, but just once in a while, they end up being adaptive. And so we're interested in that variation that helps organisms survive in changing environments. So we're studying these mutations as the hopeful monsters within the genome. So the genetic changes that may be useful, especially when you have rapid or drastic shifts in selective pressures. And if you look at my CV, people often note that I've studied lots of different things. So in my PhD, I focused on Drosophila melanogaster because it was one of the few species that had been sequenced with a fully complete reference genome that was annotated. As sequencing has progressed, we moved into non-model Drosophila like Drosophila eucuba, and we have an NIH grant where we could fund a postdoc or a student to work on local adaptation in that species. But then you can branch out, and now we can generate genome sequences for any animal we choose. So I've worked on them in ancient DNA for Neanderthals and woolly mammoths. I got dragged into these dreadful poison dart frogs that Roberto Marquez can tell you about, where we couldn't put their genome together, but we could figure out what was wrong with them. And then more recently, my lab has started working on these freshwater mussels. But across all of these different organisms, the genetic questions remain the same. So some people pick their organism and they study everything about that organism, the environment, the genome, the duplications, the SNPs, the behavior. Instead, I study these types of mutations in lots of different animals with a central genetic question focused on sources of genetic novelty. So that's my identity as a scientist, and we hope across all these projects to study how these mutations play a role in evolutionary change, for good and for bad. One of the questions that we're interested in, and one of the reasons why we need lots of different organisms for our work, is because we're interested in how population size influences the evolutionary outcomes that we see. There's a lot of literature, especially from the theory, that says that when this equation, flora times the effective population size times the selective coefficient, if that number is greater than 1, then that mutation can be seen by selection. And so you get deterministic selective sweeps, or if it's detrimental, it should get weeded out of the population. If that number is less than 1, then it will be subject to forces of genetic drift. That drift will be stronger than selection. And so you can have bad mutations persist in your population, and it makes it harder for selective sweeps to spread through your population. And especially with respect to our genome structure changes, there's a lot of literature from Mike Lynch about how genome features may be defined by these nearly neutral dynamics. So we have our organisms like Drosophila, which have very large population sizes, or Megalonitis nervosa, which is going to be what I talk about today, that also has very large population sizes and is a successful species. And so these should have lots of genetic diversity, and they should have forces of genetic drift that are relatively minimal. But we're studying these same mutations in other systems, like this Elliptiospinosa, which may in fact be extinct in the wild now, or our elephantids. We're working on Asian elephants and previously on woolly mammoths, where the forces of genetic drift are very strong. And so we can compare how the evolutionary outcomes look in these species with large population sizes and small population sizes to see what's different. Your nearly neutral selection coefficient isn't the only thing that changes with population size. The amount of standing genetic variation is also defined with respect to the effective population size and the mutation rate. So in organisms with large population sizes, you should expect to see more genetic variation than in a similar species with a small population size, assuming the same mutation rate. So how does the spectrum of standing variation in a population influence the evolutionary potential and the outcomes that you get if there's a shift in a selective pressure? And the amount of time for a brand new mutation to appear in a population will be different in a small population than in a large population. In a large population, you get more organisms that could mutate. That's like buying more lottery tickets. And so the amount of time until you get a deterministic selective sweep through the population for a beneficial allele will be shorter than in a small population where you have fewer lottery tickets, fewer chances of success, and stronger forces of genetic drift. So we expect to see very different evolutionary responses here, especially with respect to our favorite mutations, which we think are less likely to be neutral than your standard single base pair change. So overall, we're trying to answer these questions by doing the same type of analysis in bioinformatics and population genetics in lots of different evolutionary systems. All right, so I started my career of doing my PhD on Drosophila genetics and evolutionary genomics, but that wasn't always the case. So I grew up in a town called Florence, Alabama. It's right across from Muscle Shoals, Alabama. A documentary about a recording studio there, which makes it sound super cool. We never saw anyone famous really in town because they kind of hid them, you know. But it's this town on the banks of the Tennessee River. So Florence, Alabama is also famous among freshwater mussel enthusiasts, which are a different breed, because they are a hotbed of species diversity for unionid bivalves. Historically, there were over 80 species of freshwater bivalves that were found at Muscle Shoals, and now the populations are threatened and endangered for many of the species, and that population diversity, the species diversity, has dropped, right? So here we have this rich background of species diversity. So all of my high school science fair projects were on these freshwater mussels. I really wanted to do genetics and genomics and eventually evolutionary genetics, but I started my scientific career, my first publication was on freshwater bivalves. So some things happened at Muscle Shoals. Back in the day, these used to be whitewater rapids. John Ross, who was the chief of the Cherokee Nation during Andrew Jackson's time, got famous because he could navigate the waters. He could talk to the Native Americans on one side of the river and the settlers on the other side of the river. But then TVA came and dammed the river in 1918, so they were no longer whitewater rapids, and it was improved navigability and provided hydroelectric power for a munitions plant that generated nitrates, ostensibly for fertilizer, but also for the military. The consequence of this, especially in 1918, is that it limited the dispersal of these freshwater mussels and also of fish hosts that they parasitize in the larval stage. And Ortman was a natural history guy who came, and he noted there was a loss of biodiversity by 1924 after the dam was formed, that species that used to be common could not be found at all, and there were species that seemed like they were going extinct. On top of that, Muscle Shoals has a lot of industry. There's a cotton mill, there's a chemical plant, there's that phosphorus, red phosphorus pile over next to TVA that high school kids would go throw rocks at, because when you do, it explodes, and they would be trying to wrestle them away. There's a mercury spill that my friend Ashley's dad works on, and it got sprayed with DDT to control the mosquitoes that carry Vivax malaria. And then with the cotton country, there's lots of ag in town, and they get sprayed with pesticides as well. So there's a very high pollution load in this river. And one of the complexities of working on my hometown is that you don't really have citations for that mercury spill that Ashley's dad works on, right? But we know a lot of this has been documented. The freshwater mussels were in fact monitored by TVA as an indicator of water quality. So there's this very rich historic record that they have of every decade, how are the freshwater mussels doing? Because if they drop dead, that means that something's wrong with the river. So these days, there are 40 species remaining at Muscle Shoals in this one reservoir. So we have 40 different species that one could sequence and look at evolutionary genetics in a single environment. And so this is the highest species diversity for unionids in the world, in my hometown, which is how I ended up working on freshwater mussels. In North America, especially in the southeast, over 70% of freshwater unionid mussels are threatened, endangered, and extinct, as this very eloquent article said. And so this is a threat to the waterways and the ecology of the system. So freshwater mussels are benthic filter feeders, and if they go missing, you get these massive algal blooms that take over the river, deplete the oxygen, and are very bad for the fish and bad for the surrounding environment. And even if you're not the world's most environmentally conscious person, this turns the river from a place where people want to hang out and swim to something where you don't want to go poking around with the dead fish. These freshwater mussels have unusual life history and unusual features. One of them is that they have dual uniparental inheritance of mitochondria. The males inherit their mitochondria from the paternal lineage, and the females inherit mitochondria from the maternal line. And they may, in fact, be involved in sex determination, the mitochondria themselves. But the male and female mitochondria in a single species will be more distantly related from one another than if you compare the female mitochondria with the female mitochondria of another species, based on the sequence diversity. So there's something very funky going on with this, and people like Sophie Breton have been working to try to figure out what's going on with those dynamics. They have this unusual life cycle. So as adults, they're benthic filter feeders that look like any other bivalve that you would expect. They filter water from the water column, they take the algae and digest it as food. The males and females, they're dioecious. For most mussel species, there are some hermaphrodites, but they have separate sexes. They spawn the sperm into the water, and the females are fertilized internally. The females will brood the offspring in their gills, and a single female about the size of your hand can hold 1 million offspring. They become sexually mature at anywhere from 4 to 10 years old or 15 years old. And then when the offspring are mature enough, they release them into the water column where they parasitize fish. So they latch onto the fins and the gills of fish. And there was a debate when I was younger about whether or not they just latch onto the fish for transport or if they suck the blood from the fish gills and parasitize them that way. Everything that we have now suggests that they actually do parasitize the fish and suck their blood. When they mature from being larvae, then they drop off the fish. So the fish transports them to someplace else in the river so they're not close to their parents or their siblings. And then they drop off as juveniles, develop into adults, and then go back to filter feeding. So here we have this very strange interaction with fish where they parasitize fish hosts. So there's parasite-host coevolutionary processes. We have the transition from benthic filter feeding to feeding on blood over the course of their life, and lots of evolutionary processes that are worth chasing down. So they're famous among freshwater ecologists for this type of unusual life cycle with lots of interesting things to chase down. Many of the females in different species will produce lures for fish. So they have mantle modifications that look like little fish in some species, especially Lampsilus, and they flick them around to try to lure in a fish, which will either try to eat it or mate with it, depending on what it's chasing after. And then instead of getting a meal, the fish is going to get a mouthful of parasites. Other species produce gobs of Glaucidia, the offspring, surrounded in mucus in a little membrane, and it's got these markings on it to make it look like a fish. So something bigger will come up and snatch onto it, and then instead of a meal, again, it gets a mouthful of parasites. And others will simply produce these mucus nets that spawn into the water, the little gobs with the little parasites in them. So my first, one of my science fair projects and my first manuscript for my career was working with these freshwater mussels with Tom Haggerty at the University of North Alabama and Jeff Garner at the Alabama Department of Conservation. And so my job was to dissect the bivalves and get the offspring out and try to quantify how many offspring they have in their gills. And they're like little bitty grains of sand. And so we did a sampling technique to estimate how many offspring they had versus their size and their age. You can count the rings on the shells to see how old these are. And so that was my part of the study of life history strategies in Megalonias nervosa. And if you get these puppies into the paper cut, the little Glaucidia, then it hurts like the devil, and they're harder to get rid of than chiggers, which is also why my anecdotal evidence for a long time was betting on the fact that they suck the fish blood. And I don't know if my bivalve allergy is related to that or not. But these things are actually cool if you do evolutionary genetics and never cared a bit about freshwater mussels in your life, and were not the girl walking around your high school telling everyone about your science fair project, right? And that's because in this single species, we have some species in this clade that are really successful. So for this species up at the top, Megalonias nervosa, the washboard mussel, there are 80 million of them in a single reservoir. And then the range goes all over the eastern United States up to Minnesota. Then we have other species like Elliptio spinosa here, where it may be extinct in the wild, and John Ware's got DNA out of one of the last individuals that was ever seen alive. And we have at least 1,000-fold variation in population size in a clade where almost no one had made genome sequences. So in parallel, a couple of other labs were trying to do reference genomes, but I got my faculty job. And one of the nice things about getting a faculty job is that they give you startup funds, and you can blow it on absolutely anything that you want to, so long as it's science, whether anyone else thinks it's fundable or not. So I started trying to make reference genomes for these unionids. I was like, look, this is going to be such an easy project. We'll do one lane of Illumina, a little bit of long-read sequencing, and then we'll do some high-C. We'll walk out with a beautiful genome, chromosome level, and then we're done. It's going to be easy, and it's going to be great. Clearly it wasn't, but we're still making reference genomes for this clade, and the one that we made is only the second for any unionid bivalve that was ever produced. And now there are 3 that I know of. All right, so we've got this genome panel that we want to put together of species that have very large population sizes, species that have extreme population contraction in recent time, and then this species that is endangered and potentially extinct that's on the IUCN Red List. And so here we can look at evolutionary processes, especially for our duplicate genes and chimeric genes and other things like that, to see how they differ across these clades. What's going on with the most successful bivalves, and how have they adapted to these changing environments, and what's going on with these small population sizes, and does it look different at the genetic level for what's spreading through the population? So we started off with the washboard mussel, which is the one that I had worked on previously. It's a very successful species. It, in fact, increased in population sizes when others were contracting. And so if you want a measure of success, this seemed like a good place to start. And we know that we can find them. So we got one individual that we made our reference specimen, and then we also got a population genetic panel of freshwater mussels. This species was also important for the freshwater pearl industry for a long time, where they were harvested for freshwater pearls and also to make buttons with the mother of pearl from their shells. So it has impacts on industry as well. All right, so we want to look at what's going on with their genomes and see how this influences evolutionary outcomes. So I contacted Jeff Garner at the Alabama Department of Conservation. I had worked with him in high school. This is him in his natural state. I actually have trouble finding a picture of him where he's not in the water. So he goes and does the scuba diving. He does surveys for the state of Alabama to look at population prevalence. So I can find published stuff that's 5 to 10 years old that talks about how many freshwater mussels there are for different species. But that's no substitute for Jeff, who's out every single year. And he can say, we thought that species was in decline, but it's actually doing okay for the past 10 years. Or we thought that species was okay, and actually it's having some trouble now. And then along with Art Bogan, he wrote this beautiful book that's like that big, you know, on all the freshwater mussels for the state of Alabama. So he is one of the world's experts and was Alabama Conservationist of the Year several years back. Got a genome, it was harder than it looks. These freshwater mussels have been an absolute pain to get long reads out of. We've also tried Hi-C a few times, and it has failed every time with different sequencing cores, and even with people who succeeded, you know, the same batch, all the vertebrates succeeded and all the freshwater mussels have failed. But we got a genome. The M50 is only 50 kb at the first pass. We've improved that now to 100 kb, but we think we are getting most of the gene content. So it's in the same state that, say, maize was when we were in Jeff Bennett's lab, where a lot of the repeats are not fully assembled, but we think we've got a good portrait of gene content. And then with some careful bioinformatics, then we can figure out what's going on. So this is one of the longer contigs from the first pass. The little green wedges are transposable elements. You can see the genes annotated, and this particular contig has 3 different cytochrome P450 genes on it. So we hope that this genome opens up doors for conservation genomics and helps our colleagues that do the boots on the ground work if they need a genome to design primers or anything else like that. And again, this is the second genome that was ever produced, and it was a far more contiguous genome than the previous Illumina assembly that was done on earlier technology. We were able to look at genetic diversity. What is theta? What is 4 times NE times mu? The heterozygosity per site is high for Megalonius nervosa, and then we can get a partial estimate from these 2 other species where we had Illumina data and could get a partial assembly. So it looks like heterozygosity is high. It's 0.007, which for reference, Drosophila will be 1 to 2% here. So it's in that realm of extremely high genetic diversity that remains even in some of these species that have more limited ranges now. So we can use estimates to look at what the standing genetic variation is, and we think that the estimate is still relatively healthy, that for SNP-based mutations, that there's lots of standing variation that's still present in the population, especially for our focal species, and we can compare that later. But, so at first I thought, well, this is going to be a genome assembly. We'll do the assembly and annotation, and we'll release it to the community. It's going to be a resource release, and it's going to be a pretty limited paper. I even thought about just preprinting it first, or maybe we send it to you know, one of the journals that's designed for resource releases. But because I am an addict and cannot help myself, I had to take a look at the duplicate gene content. And I did this almost because I'm like, well, I got all the scripts written, it's automated, and I'll take a look. And then what popped out was a much more interesting story with respect to genetic novelty especially. So we can look at the ages of duplicate genes using a molecular clock technique. And we can look at the divergence at synonymous sites for 2 duplicate copies in the genome that are closely related. If it's been a long time since they duplicated, then you see more divergence at synonymous sites. If these duplicated yesterday, then the 2 duplicate copies should look almost identical. And so then we can fit these birth-death models that were implemented, you know, when I was a grad student, we did similar work in Drosophila, and other people have applied these models in plants. And you can get an estimate just from a single genome of what the mutation rate is for duplicate gene formation. And we found that the duplicate gene formation rate was relatively high, around 10 to the minus 8th per gene per generation, which is about what we would expect to see in a mammal, and maybe about 10-fold higher than what we would see in a fruit fly. So that's cool, that's nice. But then there were these large gene families that popped out. We did not go looking for them. They were hit you over the head obvious. There was one gene family that ended up having 143 copies in the first genome. And we can fit these birth-death models to this duplicate gene family because it has so many members. So the red line shows the duplicate gene family, its birth-death process. And then the dashed line is the same curve that I showed you on the previous slide of the background genomic levels. And we see a significantly higher duplication rate, effectively, and a higher retention rate for these cytochrome P450 genes compared to what you would expect on background levels. And to make these genome outliers, you would either have to have a mutational process that makes it more common that they duplicate, which could happen if they got put next to transposable elements by accident, or you could have natural selection causing them to be retained, and then you get an effectively higher duplication rate in your population. And cytochrome P450 genes are the classic detox genes that help fruit flies survive in polluted environments where they've been sprayed with DDT, right? And then they're also very popular in mice that have been, in rodents that have been hit with toxins, I believe morning glory, that's published? Okay. And in some morning glories that have been hit with herbicides, right? Which Gina can tell you all about. So people think that, say, mice have a lot with 40, and here we have 143 in a freshwater mussel in a river that's been sprayed with DDT since the 1950s, right? So it fits perfectly with what we know about the environment, and also what we know about the toxicity load that these bivalves can handle. We did a similar analysis for all the large gene families, and this was done computationally completely blind with respect to which functions should be present. And in fact, we were so blind that some of these functions, I could not have told you what they were until we found the gene family and looked them up. These are the large gene families that have significantly higher rates of duplication or lower rates of loss compared to the background levels. We have these mitochondrial management proteins, and they've got this strange dual uniparental inheritance of mitochondria. We have these von Willebrand proteins, which are anticoagulant proteins, and we think this is related to the fact that they suck the fish blood when they parasitize the fish hosts. We see things like these HSP70s, which are general stress formation, the cytochromes and their interaction partners, the ABC transporters, and then these chitin genes, which are important for shell formation. And at the phenotypic level, some people have suggested that this species has gotten much thicker shells over the years to help it escape predation and possibly lay down shells as the concentration of different ions may have fluctuated in the river. There's also these opsins. We have no earthly idea of what's going on with them, but we know that they're amplifying like crazy. So here in this computationally blind approach, because we were obsessed with duplicate genes, we were able to find these cases of rapid expansion. On top of that, we have 30-something duplicate genes that have dn over ds much greater than 1 on the branch where they formed. So that's a strong signature of selection, and we think that duplicate genes have been extremely important for adaptation to changing environments. And again, what we find in the genome is recapitulating what we know about the biology of the organism, which is reassuring. So, you start your PhD, and if you had told me at the beginning, you asked me, like, what are you going to find in your thesis? I would be like, oh, duplicate genes do everything. They do pesticide resistance. We're going to find developmental innovation and all that. And then you actually do your thesis, and it's more normal than that, but you still find exciting things. This is the genetic daydreams hanging out in these freshwater mussel genomes that I had imagined working on from a genetics perspective when I was much younger. So that's been really exciting to see. One of the questions we got, especially from the reviewer, is Megalonias nervosa special, right? It is not necessarily special for a freshwater mussel. There were transcriptomes available, not whole genomes, but transcriptomes available, where we can compare the number of detox genes in Megalonias nervosa to other freshwater mussels, where we had just FASTQ files that were either in the SRA or were incidentally collected by collaborators. And so if you look at the cytochromes and the ABC transporters, M. nervosa in green here is actually quite modest. And also with these thioreductions and the glutathione transferases. But the number of transcripts that they have is monstrous for these detox genes in these filter feeders that have faced environmental changes. So at the transcriptome level for multiple species, we see a similar process. So we think that the freshwater mussels have turned on a Xerox machine and copied their genes as a way of adapting to new environments. They've got lots of gene copies and then selection acts on them, but the amplification of their genes is one way that they adapt to these changing environments, and that those are spreading through the population and a very important aspect of how they're adapting. When we see duplicate genes go crazy, one of the first questions is what's going on with your transposable elements, these selfish genes that copy whether it's good for the host or not, and they oftentimes create new copies of duplicate genes when they move around the genome. We can look at the TE content. and compare it to 3 different species where FASTQ files were available. We do this in a reference genome-free annotation pipeline from Rep de Novo. It was created for a poison dart frog genome that we couldn't put together. We can apply it here to these freshwater mussels. Venusticonca ellipsiformis from Sophie Breton's group has a very high palintin element content. But in every other class of transposable elements, Megalonias nervosa has more recently copied transposable elements. And in all, it has 500 megabases more sequence of recently copied TEs compared to the other species that we have available. So it's had this expansion in genome size, this activation of transposable elements, and also lots of duplicate genes. From what we can see now with the population genetic data, most of, and possibly all of those TE amplification cases, we don't have any good candidates for adaptation. So we think the duplicate gene amplification is adaptive, and the TEs generally are not adaptive. So here we have this portrait of genetic novelty in this freshwater mussel, and the ways that it's expanding. So there's some information that we can get from a single genome, but to get really precise views of adaptation, we would generally go to population genetic data. And so we want to be able to sort out what is due to natural selection and what is due to genetic drift with greater precision, right? And so we search for signatures of selection in our population, and the 2 signatures that we look for are reduced sequence diversity throughout the population. So it's like you walk into school one day and everyone's wearing like pink pants, right? Because that's new and cool and different and everybody wants to have it. The same thing happens with your sequence diversity when there's a mutation that's favored by selection. You look across the population, everyone has the same thing because that gene has spread due to natural selection. And it drags along the linked variation at adjacent sites. The other thing that we see are skewed site frequency spectra. So normally in a population, you'll have some mutations that are found only in one individual, some that are found only in related individuals, some that are found in lots of people throughout the population and are at medium frequency, and then a few that have spread through the population all the way to fixation because of genetic drift. When you get selection, it will drive out the moderate frequency variation and give you more rare stuff and more common stuff than you had before. So we can do these genetic scans for selection. Your average contig in this genome that we've improved to an N50 of 100 kb, we have, I think our longest contig was a megabase long. We have the distribution of sequence diversity is shown with this density plot to the right of the graph, and the dashed line is the genome-wide background rate. So we can compare these distributions. This is a fairly normal contig, but we do see these regions like this that show dips that are consistent with very strong selection. And we can also look at TGMAS-D, which looks at the site frequency spectra, and if it dips as well, then those are the signatures that we would normally want to see if we're doing selection scans. So just to put a background on this, these are the 2 best in show selective sweeps in the Drosophila genome, what's considered very strong selection from my thesis, where population geneticists really like to see Vs, and we like to see genetic diversity go down to 0, and that's one of our signatures of selection. When we look across the Megalonias nervosa genome, we saw some sweeps that look like that, but we also saw this. So genetic diversity absolutely flatlines for up to a megabase in this region. And so we've used another genome from Chase Smith at Texas to scaffold these pieces of the genome together. But there's almost no genetic variation across the entire population here for up to a megabase, which I have never seen in my entire life. Like I almost fell out of my chair. The first thing you should ask when you see something that you've never seen before in your life is it an artifact? When we look at sequencing coverage, it's normal. When we look at the contigs, we see no sign that it's misassembled in terms of splitting haplotypes. We looked at gene sequences on here, and so far I have not found a bioinformatic artifact that could cause this. We think that the genetic diversity across the population has actually been driven out completely, which would be consistent with extremely strong, very recent selection in this population. And of course, it's in a species where we know that they've gone through ecological upheaval, and they've had very strong selective pressures at the phenotypic level. So these selective sweeps actually are so extreme that they don't fit the population genetic models that we would normally use to estimate selection coefficients. and they are consistent potentially with lethal selection, a lethal trait. As a population geneticist, this is about the most amazing signal that I've ever seen in my life. The first question, of course, is it drift? So we did, first I checked to see if there's some sort of bioinformatic artifact. I have not found one after staying awake at night a lot, worrying about it. We can do simulations to ask what would happen under different scenarios if there were demographic changes. If there's a population expansion, a population contraction, what would happen to genetic diversity? We can even lower the recombination rate, which you can see here, from 3 megabases over to 5 megabases, which increases the variance, but it doesn't produce anything like what we see. It can produce dips, but they're not as extreme as what we see. They don't go down to 0. And they're not as broad and as wide as what we see in our natural population. And we did lots and lots of simulations. There's 15 different graphs that we put into the supplement. And there's more that we actually did that are ridiculous scenarios. And there is no scenario that I can come up with that would cause these signals in the genetic data without also wiping out genetic diversity genome-wide. So genetic drift usually does not cause one section to be an outlier with respect to the rest of the genome, so much as it increases the variance. We also had someone ask, what would happen if you had all of your freshwater mussels, like after the dam was formed, die off, and then they got replaced by a very small population and there was this extreme bottleneck? We could have 80 million individuals go down to 100 individuals. And based on the population genetic equations, we should still have 90% of our heterozygosity left. It won't take it down to 0. There's only so much that genetic drift can do in a short time period. And so genetic drift cannot cause the genetic diversity to go absolutely down to 0. And again, it would affect the genome as a whole, not just one region. The other argument, I'm going to give you this picture instead of what I'm actually going to talk about. So after working on fruit flies, there aren't very many fossils for fruit flies, right? They're little insects, and you squish them, and there's not much left. And then if you wanted to find them, they're really small, and paleontologists aren't so into fruit flies, I think. So, there's not a lot of fossils that are known. So we talk in Drosophila about how to model population genetics. And there are all these unknowns. Was it a population expansion? Was it a population contraction? These freshwater mussels leave shells behind. They're also found in the shell mounds that were left by Native Americans, and they've been surveyed there where they can get rough estimates of species diversity long in the past. And then in historic time, they've been monitored in this exact river by a government agency that went out and counted the mussels every year, where you can go and get those records. But we had an issue in shipping where a couple of our mussels didn't make it, and then they reeked to high heaven. And then there were a couple of mussels that were not doing great, but they were still a little bit responsive, and I had the brilliant idea to stick them in a bucket from my fish tank over the weekend, and they did not make it, and we had to throw away the bucket, and I had to apologize to everyone on the floor for the smell. If 80 million freshwater mussels died off, you would know, right? It would be ecological upheaval. Everyone in town would know about it, and it would be a horrible disaster. We have no record of that happening. It's not consistent with the historical or paleontological data from the shells, right? So we can rule out certain aspects of what kinds of bottlenecks are possible. But again, even if it did happen, it wouldn't produce what we see, right? So that's one of the benefits of working in this system, right? So is it genetic drift? No, I'm sorry. We have not found any scenario that would cause the signals that we see via genetic drift. Here's what happens with very strong selection. So the simulators won't allow us to simulate a purely lethal trait, but we can crank up the selection coefficient. In a 1 plus S model, we can take it up to a selection coefficient of 100. If you're used to the 1 minus S selection coefficients in your population geneticist, that's equivalent to 0.99. Right? So a 1% chance of survival if you've got the wrong genotype. Then we start to see these massive dips in genetic diversity, but it goes back to normal levels at a few megabases away. And so the spread in these signals starts to go down to 0 and gets wider and wider. And we're sort of at the limit of what we can do with these simulations, but there is nothing but selection that I have found that can cause these types of signals in the genetic data that we observe. We also went back to our gene families that had expanded, and we have some of these show signatures of selection. Some of them show signatures of selection long in the past for an ancient duplication, but there were dozens of copies that show dn over ds greater than 1 on the terminal branch, and were also located in regions that had signatures of selective sweeps. So that's 2 independent measures of adaptation and 2 independent measures of selection that we don't necessarily think would have to be coincident, but 2 different analyses that are telling us this particular gene and this particular gene duplication was adaptive. So we have some of our favorites where we have a gene where there's a known function. There's a furin gene in this region. The purple is an annotated gene sequence. The green is a transposable element. Gray is something that the gene prediction software finds but doesn't necessarily have any other support beyond the computational prediction. In this region, the only gene that is present is a furin. It's an anticoagulation gene. That fits again with the fish hosts and the evolution with blood sucking. Right, so it ties back to the biology of the organism. There's a chitin synthetase that we think is important for cell formation, and it again is the only gene that's found in this region. And it has skewed site frequency spectra shown with the G-D, and it also has skewed genetic diversity. At the bottom, the coverage across this region is normal, except for a couple of repeats, but they're not the region that we think is swept. And you can see the density plot of these, where there's even a bimodal distribution into G-D. There's the normal parts of this contig, and then there's the region that shows the selective sweep signal showing up as the other hump. And then last one, a cytochrome P450 gene, where there are 2 copies here of cytochrome genes in this region that shows a signature of a selective sweep. And unless there's some other unannotated thing, then there's no other candidate for selection. So that we can nail down. On top of that, we find some regions where we see a selective sweep. We're working with a genome. Its closest relative that was annotated and functionally characterized is 200 million years divergent. So this isn't as easy as, say, annotating a new mammalian genome or a new fruit fly genome, where you have lots of function to go on. It's difficult to match them up in a BLAST-P, and so we do get some functional information, but we're probably missing a lot. And our precision here in nailing down may be limited. One of the questions we get is, was this a modern selective sweep since the dam was put in and since DDT got sprayed on the river, or is it historic? We don't know. It's as recent as you can get from population genetic signatures, but our resolution on how far back in time is a little bit limited. So it could be, we just don't know. It could be that it happened in the past 15 generations since the dam was formed. Or it could be much further in the past, like 2,000 years ago. When you start to get to 5,000 or 10,000 generations, that's too far in the past. But we know that this was recent in evolutionary time. Historic DNA might help, and so we're talking to Christine Evers, who has a working protocol to get DNA out of shells. I was working on something similar before the Dean clawed back some of our flex money during COVID, and it's a little bit more challenging than getting DNA out of bones, which I'm happy to talk more about if anyone's interested. One of the challenges is that if you dissolve calcium carbonate, it produces carbon dioxide, and then your tubes explode. That doesn't help. So Christine has solved these problems mostly by using less shell powder at once and then combining them at the end. So we're hoping that in the future we can start to tackle those questions using some museum collections if we can persuade the curators to give us access to the samples. So right now we've gotten some money from UNCC to keep working on our reference genomes. Our goal is to do up to 15 species of bivalves that are sitting in the freezer. So we want to take this evolutionary system to the point where Drosophila was when I was in graduate school, where you have reference genomes for multiple individuals, multiple species across your clade, and so that we can expand the genetic resources, especially for those that have relatives that are endangered or extinct. And we can look at our evolutionary genetic questions of what's happening when you change population sizes. We made a phylogeny because we could. That's your extra slide. Okay. So now we have these freshwater mussels, and we can answer these questions of how does evolution change. So we've got some in our panel, like Megalonitis nervosa and Pentamelus elatus, that are millions of individuals in one reservoir, broad geographic ranges. They are in no danger of going extinct. We have things at the other end of the spectrum that have gone through extreme population contraction and are a little bit difficult to find. And then we have this species, Elliptio spinosa, which I am really, really excited about. So John Ware's at the and so we can start to look at genetic diversity. So John Wares at UGA heard that I was working on Unionidae. So I met him when I was an undergrad at Georgia, and he was just being hired. He said, I have DNA in my freezer for a species called Elliptiospinosa. It's now on the IUCN Red List. There were surveys where the last individuals that have ever been seen alive were in 2005 and in 2007. There were 8 individuals seen alive in 2007. From the specimen in 2005, they extracted hemolymph, so that they got the equivalent of a blood draw from a freshwater mussel, and then they put it back into the river. And because John Wares did an old school phenylchloroform extraction instead of a QIAGEN kit, the DNA is intact enough that we could get long read sequences. So we have a reference genome now with an N50 of 260 kb. We still have some DNA left in the freezer. And we may try some more sequencing for this. And we have a reference genome for a species that may in fact be extinct, which is about as cool as it gets. Like, I can't believe, like, all the coincidences that had to happen, that this person had to know me, had to be willing to send it, had to have done the right DNA extraction in the past, had to have had access to the specimen. And so we're hoping we can compare it to one of its nearest relatives, Elliptiocrassidens, that was already in our genome panel. So we're putting together a proposal with Christine, who again is the one who's gotten the shell extraction protocol. So they have 50 plus specimens from the 1950s for this species at the Georgia Museum of Natural History. And then 10 specimens that they have from the 1970s when it was becoming more rare, and a few from the 1990s, and our reference genome from 2005. There's no DNA from the last 8 individuals because they decided it was not worth perturbing them and causing any damage to them. But we're trying to see if we can get funding to do historic DNA and look at the time series of what's changing in their genomes as the populations are dropping off. Do they also have an expansion of detox genes, or is that completely missing? Is that part of why they went extinct or not? What's going on with the genetic diversity, and how quickly does it drop? Are these things going extinct too fast for the genetic diversity to drop off, or is genetic diversity also going missing right as they need it to adapt to changing environments? So that's one of the questions that we hope to be able to answer in the future, and it's really, really exciting. All right, so with that, I'd like to thank all of my collaborators, especially Jeff Garner, who ships us all the samples from Alabama and has been outrageously helpful. And he's also put up with me talking about population genetics at him, some of which, you know, he's a fabulous scientist, and he knows all the things about these freshwater mussels and is one of their greatest advocates. All right, so I'm happy to take questions, including things about assembling genomes that are a little bit difficult to work with. Yes?
Do you know if there's anything weird going on with the combination that might cause like a really long region to be really swept?
Right, so is there anything going on with recombination? Like, could there be an inversion making this region swept? There could be a locally suppressed recombination thing that would extend it out a little bit, but it's not that recombination is broadly reduced. And we think even without recombination being suppressed, you can still get these large selective sweeps. But that's one of the questions. And one of the frustrating things about having a genome that's so fragmented is that we can't look at my favorite mutations, the chromosomal rearrangements. We can still get duplicate gene content, but we can't look at rearrangements per se. It would be really nice if we can start to sort that out as the genomes become more contiguous. But it definitely takes selection, even if it's selection plus something else. Yeah, good question. Yes, Gina. Right.
I mean, they might have some sort of resistance mechanism too. Is it necessarily the blood sucking? And right, because anticoagulant makes you think like warfarin and others. I mean, you showed multiple selective agents happening.
Right.
Right. Multiple chemicals.
Yeah.
Impacting it. So I'm thinking like, is there any potential that buildup of chitin could actually help the muscles not get exposed to the chemicals? Is it necessarily?
Is it the thing that we're saying it is for the selective pressure?
Could all these things be working together to help it with all the nasty chemicals that the muscles are being exposed to, right? So
right.
Not just detox, but it's like...
Yeah, if they interact together. So the question was, for those online, with things like the coagulants or some of the other proteins? Could it be that they all interact with detox instead of, the fish host and stuff? We know that the loss of the fish host is a massive selective pressure. But yeah, I mean, there could be room for a hypothesis that some of the toxins cause bad physiological problems that they're adapting to. I guess one of the questions would be to find a reservoir where they're hit with fewer toxins and see if you still see the adaptation to the fish hosts. We know that there has been fish host switching in these species because of just the change in the fish levels. So Megalonias nervosa is a generalist, and it feeds on carp and catfish, which are in the river all the time if you go fishing there. And then there are other species that are in much worse shape that are specialists on very specific fish. So we think there's a little bit of storytelling, but the way that we found these was through the computational stuff. It does fit with the biology of the organism. We tried to phrase it carefully to not go overboard, but yeah.
Just a quick follow-up. Are these, I don't know how to hold this. Okay.
The cube.
So is the life cycle fast enough? And I think it is, that you could actually do selection and start to parse away some of these enzymes that could be, these proteins that could be involved in adaptation.
What do you mean by selection?
You could do
experimental evolution?
Yeah, do some experimental evolution.
The generation time is 10 years.
Okay.
It's 10 years to maturity, and so the generation time is probably 30 years because they can live to be, there are some claims of 60, but a 56-year-old mussel, which is gigantic. So their life history looks more like a human once they mature, but it's sort of like some of these plants that produce tons of seeds. They produce tons of glycidia. That's a main point of attrition. So some people will rear these in captivity to get them onto a fish host for an endangered species and then drop the adults into the river because they're much more stable. But we have a generation time problem. We will not be CRISPRing them because no PhD student is signing up for a 10-year PhD. Yeah. Or they shouldn't if they want to. Yeah. Mo, you have a question?
So you found a lot of genes, or a lot of duplications that seem to have undergone development. Does this genome just produce more duplicates than others as a consequence of being in a highly stressful environment?
Right. So does this genome produce more duplications than you would expect just because of the stress in the environment? That recent burst of transposable elements could be due to recent stress. Some of the duplications are ancient. It produces about the same per gene duplication rate as a mammal, and its genome structure looks more like a mammal. It's a 2 gigabase to 3 gigabase genome. The TE content has a few more recently proliferated TEs than a mammal, but it looks similar in terms of genome content. So I don't think the background duplication rate is necessarily elevated. These cytochromes, on the other hand, on top of the ones that we used for our gene family study, there is this strange fragment that has part of a cytochrome and part of an ABC transporter. And they're the sections that interact. So they're not the full gene, but part of it. And then I think for at least some of the copies, they've got TEs on either side. That thing is in the genome so many times that BLAST stops printing out hits. So there's like hundreds to thousands of these things in the genome on top of the full gene families that we're working with. And that cassette has been copied over and over and over again. That could be activated by stress and then produce something that seems like it's related to detox, but what would it do? I don't know. We've got more questions than answers there. There's something going on with that cassette that is very mutation-driven. With the others, we think that there's definitely, they've been amplified more than you would expect based on the genome background, right? Yeah.
Cool, thanks.
Thanks. You can tell a department that's used to thinking about evolutionary genetics on the questions. Yeah.
I think we had a question in the chat. If
Okay.
We have a second.
Yeah, I can't see it.
Do you observe any within species polymorphisms in copy number for your family of duplicated genes?
Do we observe within species copy number variation for these genes? We do on some. We haven't totally nailed down some of the polymorphism because of some of the bioinformatic challenges. So that would be the dream if our genome were slightly better assembled. We sort of left that alone for the most recent publication, but I would really love to come back to it that's one of the goals when we submitted a proposal to the NSF. So we would like to come back to that with these more contiguous genomes that we're getting. Yes?
Before you ask the question.
So we know that onionids exhibit this really spectacular clinal variation from headwaters to downstream phenotypically. So for the population genetics component of your work, I was wondering how spatially far apart were the individuals and if they were undergoing different kinds of selection.
Right, so given some of the phenotypic clines in bivalves, then how far apart were these individuals sampled? Jeff got them all on one dive from one reservoir, which was our goal because we wanted to keep the environment constant. So all of our species we're getting from the same reservoir. So from a population genetics perspective, that controls your variables that are present. So we can look at evolutionary processes without worrying that like some other reservoir got hit less by pesticides. Some people are really interested in how different species are from one reservoir to another, and some species show more local variation. Megalonitis nervosa has a really wide range. These probably could hitchhike along barges along the river. Darwin's last paper, which I gratuitously cite in my grant, is on how he saw a unionid bivalve latch onto the leg of a bird and get transported from one reservoir to another. We don't know how much they spread along the river or how much the migration might be. There's some single gene evidence that perhaps the megalonitis in Mexico may be very different from the southeast, but I think they're more mixed for this species. Yeah. There's only so many questions I can do at once, and I've told myself no new projects, but that one has been suggested multiple times and may be on the list later. And once you get your reference genome, then it's one lane of Illumina to get a population, right? Like doing the extra studies downstream, that's way easier once you get your reference genome. I'm recruiting students on fruit flies where I have money, but really I wish we could chase down this with an army of people. Thank you.